Lesson 1I, Proteins. All of these foods are protein-rich. Proteins are large, complex molecules that have many important structural, functional, and regulatory roles in the human body. Some proteins in the human body include antibodies to fight infections, enzymes to carry out the thousands of chemical reactions in the body, hormones like insulin, and muscle and hair fibers. Proteins are polymers of hundreds to thousands of monomers called amino acids. There are 20 different amino acids that can be joined in nearly unlimited combinations in various lengths to create the thousands of different proteins required for life. Each of the 20 amino acids have a common amino group and acidic group, but has a unique remainder group, also called the variant group. This remainder group is what distinguishes one amino acid from the other 19 that exist. Here we see two of the more simpler amino acids, glycine with a simple hydrogen as its remainder group, and alanine with a methyl group for its variant group. On this slide we see three more amino acids, valine with a branched carbon variant, cysteine with a sulfur in its variant group, and phenylalanine whose R group is a hexene ring. Here is the complete list of the amino acids grouped by their polarity and charges. When amino acids are polymerized into a single long chain, we call it a polypeptide or a polypeptide chain. This bonding of amino acids is achieved through a dehydration synthesis reaction, where two amino acids are joined together with the help of an enzyme and a water molecule is produced. Here we are joining glycine with alanine to create a dipeptide. This video illustrates how amino acids are polymerized together using a dehydration synthesis reaction, creating a peptide bond. For a functional protein, its final shape is very important. An error to the amino acid sequence can lead to a final structure that could affect the protein's function. It could make the protein function worse or possibly better, which is the root of evolution. Of course, there may be no change in its function either, if the error is slight. The final shape of the protein involves a long chain of amino acids, called the primary structure, to coil up or line up in sheets to form something called the secondary structure. The coiling on the left is called alpha helixing, the folding on the right is called beta-plated sheets. The secondary structure of coiling and folding is caused by hydrogen bonds. The next stage is where the secondary structure, with its alpha helixes and beta-plated sheets, clumps up due to covalent bonds, which creates a globular structure. We call this next structure, that sort of looks like a fist, the tertiary structure. The final structure is called the quaternary structure, and this is where two or more globular structures come together, joined by H bonds to form the functional protein. Remember, any variation to the final form can cause the protein to function differently, for better or for worse. Here we see the primary structure, which is simply a long chain of amino acids called a polypeptide chain. With alpha helixing and beta-pleated sheeting, we get the secondary structure, formed with H bonds. Then we have the tertiary structure, where the secondary structure fists up or balls up into a globular structure. The quaternary structure is where two or more globular structures come together and are bound together by covalent bonds. This final structure is very important to its function. Here is an example of the final quaternary structure of hemoglobin, the important blood protein responsible for carrying oxygen from your lungs to the different tissues of your body. It has four tertiary structures, and each subunit carries an iron-containing compound that binds oxygen and turns your blood a bright red. In order for humans to build these proteins, we need to ingest protein and digest them into their monomer amino acids. This enzymatic reaction that breaks peptide bonds is called hydrolysis and is illustrated by this video clip.